in Australia. Good morning, everybody. So I'll start
Good
to
morning.
talk my presentation by movie file. So just a moment. Can you hear? Can you look my movie file?
Yeah, we can hear you,
Oh,
and we can see the present slide as well. Yep.
adjustment. I have abandoned adjustment. Yes, I will start.
Yes, sure.
Thank you, Chairman, for inviting me to this conference. Today, I shall talk about brain activity underlying olfactory gustatory synchrony perception. Just a moment. Can you hear my narration?
Yes, I can hear you very well.
Oh, okay, thank you very much. So I will
Thank you.
continue. Using
Yes, sure,
event
please.
limit potentials.
J. Gibson, who put forth affordance theory, said that the psychological origin of sensation is less important than that the sensations can be used to identify objects that are biologically crucial in the case of odors and tastes. Then, what sensation is identification objects that are biologically crucial in case of taste and smell? One of these situations is food perception. In Pragnant's law in Gestalt psychology, there are some important laws for human cognition process. In this presentation, we focused two laws from this Pragnant's law for food perception. One of these laws is law of proximity. This law states that when an individual perceives an assortment of objects, they perceive objects that are close to each other as forming a group. And the other law is law of similarity. This law states that elements within an assortment of objects are perceptually grouped together if they are similar to each other. The keyword for low proximity is close to each other as forming a group, and for law of similarity is similarity. Then we have to reinterpret these keywords close and similar in food perception. In food perception, close would mean temporal simultaneity in taste and smell stimuli. And then, what is similar in food perception? This would be familiarity of combination between taste and smell stimuli. Familiarity of smell and taste combination means how frequent we have experienced this combination of smell and taste in daily life. Based on these ideas, we performed psychological experiment simultaneity judgment task between taste and smell stimuli. We tried to use taste and smell stimuli, soy sauce odor, and sodium chloride solution respectively for familiar condition. Coumarin, that is smell of cherry blossom leaf, for smell stimulus, and sodium chloride solution for unfamiliar condition. From now on, I shall show you example of this simultaneity judgment experiment. Please imagine blue circle as taste stimuli and pink circle as smell stimuli. you will see taste blue circle, smell pink circle appear at the same time. This case, taste and smell presented to participants at the same time physically, and almost participants will feel simultaneous taste and smell stimuli are presented. In this case, first taste is presented. 500 milliseconds after taste presentation, smell stimuli is presented. In this example, you will clearly perceive these stimuli are presented separately, say, not simultaneously, by visual sample. But how do you feel in case of real taste and smell stimuli? In this case, first smell is presented. 500 milliseconds after smell presentation, taste stimulus is presented. In this example, you will clearly perceive these stimuli are presented separately, say not simultaneously, by visual sample. But how do you feel in case of real taste and smell stimuli? For precise presentation of smell and taste stimuli, we used olfactory and gustatory stimulators for event-related potential measurements. Those stimulators can realize pure stimuli without touch sensation, that is, under sensory threshold pressure change, and realize enough steep stimuli rising time, optimized for ERP or reaction time measurement. By gustatory stimulator, tip of participant's tongue covered a small hole on the wall of Tygon tube, so liquid did not flow into inside of mouth. And we performed real-time monitoring with sub-millisecond order, using ultrasonic gas sensor for olfactory stimuli and photo sensor for gustatory stimuli. Experimental condition for taste and smell stimuli like this. This means, first, smell stimulus was presented to participant. 600 seconds after smell stimuli, taste stimulus was presented. Then, participant was asked to answer which do you feel this smell and taste combination came at the same time or separately. Participants answered using their fingers. After 300 smell and taste combination presentations and judgments for various time shift conditions, we calculated probability distribution like this. Vertical line showed rate of answer of same time from total answers from each time shift condition. For example, this point means that in the case olfactory stimulus came first and 450 seconds after taste stimuli came, participant answered taste and smell combination came in the same time with 40% and came separately with 60%. Left figure shows probability distributions of vision and audition from other researchers' result. And right one shows that taste and smell, that is our original data. You see the bell shape of right figure is broader than that of left one. This means human can distinguish combination of visual auditory stimuli combination with high temporal resolution than that of gustatory and olfactory stimuli. Right figure is result of familiar combination, soy sauce flavor and sodium chloride, and left distribution is result of unfamiliar combination, coumarin and sodium chloride. In familiar combination, probability distribution showed significantly broader than that in unfamiliar combination. In previous slide, I said in familiar combination, probability distribution showed significantly broader than that in unfamiliar combination. This result means in familiar combination, that is, frequently experienced in daily life, showed lower temporal resolution than novel one. This phenomenon would imply that familiar combination of smell and taste forms tighter flavor object. In other words, according to this flavor object, taste information and smell information bind together intensively. This would suppress or weaken onset of taste or smell onset. This mechanism leads lower temporal resolution in discrimination between taste and smell. We therefore, in order to investigate this hypothesis, we measured event-related potentials of gustation and olfaction. This figure shows olfactory event-related potential for three conditions. Green line shows averaged potentials measured in another experiment. That was simple event-related potential with no task. Blue line shows averaged potentials in the case that participants judged that smell and taste stimuli came separately, and red one shows participants judged that smell and taste stimuli came at the same time. In short latency, we could not find significant difference. This figure shows gustatory event-related potentials for three conditions. Green line shows potentials measured in another experiment, simple event-related potentials with no task. Blue line shows averaged potentials in the case participants judged that smell and taste stimuli came separately, and red one shows participants judged that smell and taste stimuli came at the same time. About 130 milliseconds after taste onset, we found significant difference between separate condition and at the same time condition. In the same time condition, the amplitude of red line was significantly smaller than that of separate condition, and the amplitude of separate condition is almost same as that of simple stimulation showed by green line. This positive component is gustatory related potential is called P1. We found significant difference in P1 amplitude. P1 amplitude when participants judged at the same time was significantly smaller than when judged as separately. Then, what is P1 component? From previous studies, this component is the first gustatory activation in the cortex, measured by EEG or MEG. The location of this activation is the transition area between the insular cortex and the central or Lowlandic operculum, or the primary gustatory cortex in human. So we conclude significant amplitude difference in the primary gustatory area in short latency. This means neural activation related to simultaneity judgment should occur between taste stimulus input and the primary gustatory area. We therefore propose this connection model. In order to affect the primary gustatory cortex, the interaction should be before the primary gustatory cortex. In the thalamus VPMPC was the neural relay for gustatory information, and about olfaction, after the piriform cortex, the information was set to project to the mediodorsal thalamus. These VPMPC and the mediodorsal thalamus may have mutual interaction through thalamic reticular nucleus. This area is said to have gating function. As a result of this gating function, the onset response in the gustatory cortex will be suppressed. This neural anatomical model is supported by the shift of point of subjective simultaneity. Summary of this presentation. Temporal resolution in simultaneity judgment became lower in familiar condition than in unfamiliar condition. This phenomenon would be caused by decline of onset response in the primary gustatory cortex. This decline of onset implied some interaction, gustation, and olfaction in the thalamus, not only in the higher cortical areas. Thank you for your attention.
Okay, thank you, everyone.